And if you look at where this maps in the embryo, it's actually just below or closer to the vaginal region compared to where gastrulation initiates and forms this dorsal blastopore lip. And so now I want to tell you about a very, very famous experiment in developmental biology by this pair of researchers named Hans Spemann and Hilda Mengel. And they were at the University of Freiburg in the early 1900s. Hans Spemann was the head of the lab and Hilda Mengel was the graduate student. And these experiments that she did led eventually to the Nobel Prize. So she's one of the first people whose dissertation as a graduate student, as a PhD student, led to a Nobel Prize. Unfortunately, she was killed shortly after these experiments were done because she had a gas heater in her apartment that exploded and killed her. So it was really sad. So Spemann and Mengel.
So she never even got to receive the award?
So she never got to receive the awards.
Yeah.
I know, harsh. So here's the experiment, and once again, it's going to involve a donor and a host embryo and a transplantation event. Only this time what's being transplanted is a little portion of tissue that is just beginning to invaginate in a very early gastrula stage embryo. So an embryo that's just starting gastrulation. She went in with a knife, and this is technically kind of tricky, cut out that little portion of the embryo that was beginning to gastrulate and moved that over to a host embryo and kind of stuck it in on the presumptive ventral side. And then waited to see what would happen with these embryos. So that's just a little bit of red coloration there to show you the transplanted cells from this donor embryo. And amazingly enough, what happened was that second site of dorsal blastopore lip continued to function as a dorsal blastopore lip. And it initiated a second site of invagination, or involution, within this embryo. So over here on the presumptive dorsal side, we have the endogenous one that started on its own and keeps on going. But then this transplanted one did the same thing. It began moving cells inward into the blastocele cavity as time went by. And you ended up with, actually now, instead of an embryo that just had an overrepresentation of dorsal fates in the form of two heads, now we get something that looks more like a Siamese twin. You get two full embryos with head and tail, dorsal and ventral, just sort of stuck together at their ventral locations. So in terms of patterning, which is the theme of the day here, this is a considerable advance over what the new Coop study was able to do. The dorsal blastopore lip had more inductive ability for this embryo. And I want to point out a couple of different things here. First, if you slice through these embryos and you look at the tissues that are developing in a cross-section, these donated cells have the ability to incorporate with and to organize the endogenous cells. So this little circular thing here, this is the neural tube. It's a precursor to the spinal cord. It's actually a tube that runs along the entire anterior posterior length. And it's one of the next or first kind of organs that will be organized within the embryo. We'll talk about it more later, but I point out here that a neural tube got organized from these donated cells. And then side by side, there's some other structures in here. It's a combination of host cells and donor cells that just integrated together and formed new pattern and new structure, apparently due to some signals here from the donated tissue. So this must mean that the dorsal blastopore lip really has some important signals for setting up dorsal ventral and probably also anterior posterior cell fates within the embryo. And this term here, primary embryonic induction, this is meant to indicate that this full dorsal axis, including a neural tube, could develop. So not just a head, but the entire body length. if you look at some of these embryos and you use markers to denote the neural tube, it can help you understand what the patterning of these things is. So here is, sort of an imperfect double embryo together, but patterning-wise, if you look at this marker for the neural tube, you can see that the two embryos have been formed here, so two complete neural tubes. So even though it's not really a perfect second embryo, patterning-wise, it's gotten a long ways. Okay. Here are two embryos. It's probably one of the best examples from this situation, but you can see actually all the way along their anterior-posterior axis is a duplication of tissue. So, technically this is difficult. You went in and cut out this little tiny piece of tissue, stuff it over in another embryo, you expect that thing to survive and also be patterned. But this shows you the potential that is there if you are able to fully integrate that donated tissue. Okay, so they call this thing the Spemann organizer. I guess Spemann got to be the one because he was the head of the lab. So these are the points I was emphasizing, that it can induce host tissue to change cell fate, become part of the second embryo. And together they are organized in a way that it establishes an anterior, posterior, and a dorsal ventral pattern within that new embryo. So now we have then a Spemann organizer and a nucleus center to contend with. And we would like to know something about what's going on between these two, what's the relationship between these two, and especially what are molecular signals that might be the key players for this. And as time goes by, we not only want to know kind of big picture things like where are the signaling centers, but we want to know the nitty-gritty of which genes are involved to cause this and what is the mechanism. So how are the genes performing their function in the embryo in order to cause an event to occur? Okay, so towards a model for explaining mesoderm induction, some of the things that we had talked about is that you have to have some sort of signal from the vegetal cells to induce mesoderm to occur. And then you also have to have something for explaining the regional polarity. And this nucleus center seems to be needed as well to provide the information for dorsal fates. So an information for mesoderm and an information for dorsal fates. At least two signals. And now we'll go ahead and move right into the next lecture, which is going to continue some of these themes. We're going to be talking about the next stage after gastrulation, which is called neurulation. This is a stage where we form the neural tube. And this is just a good place to introduce a commentary on cell signaling here. There are 4 different kinds of communication mechanisms that we're going to see a lot of in developmental biology. So I'll just go ahead and introduce what they are. First would be transcription factors. There's a great deal of very specific transcription factors that have important jobs to do. And you can think of this as communication within the cell. So we've talked about macho-1 already with the sea urchin. And later we'll talk about homeodomain proteins. And these are in the nucleus regulating differential gene transcription. Then paraffin factors. These are not in the nucleus. In fact, it's the opposite extreme. They're actually excreted from cells, and they're used as short-range signaling molecules within the embryo. So short range would probably mean from, say, 1 to 10 cells away in general. So kind of the immediate neighbors. You don't actually have to be touching, but you have to be in the vicinity. So communication to nearby cells. And so far we've talked about WNTs, and we're going to talk about TGF-beta family here in a minute. And then juxtacrine factor. This is also a cell surface method of communication, but it really requires the actual physical contact of 2 cells. So 2 neighboring cells, and these kind of communications often involve membrane-bound proteins, either transmembrane or stuck in the membrane, that are not secreted. So they can't get 5 cells worth away. It has to be just the neighboring cells that are immediately next door. And then, since you've heard of this before, endocrine factors like hormones coming from the brain, say the pituitary, and influencing all the way down to the reproductive organs, those would be endocrine factors. So they're a long-range signal within the body, far greater distances than 10 cells worth. So with these 4 sorts of communication molecules, we can accomplish different goals within the embryo. Okay, so paracrine factor, we're going to start talking more about, and just some examples of this. Here's a little visual of one cell secreting some dots, which are paracrine factors, over to the neighbors. Wind is an example of this. It's a small molecule that's secreted and may influence neighboring cells. Other members of the family are anything that ends with growth factor. So epidermal growth factor, EGF, fibroblast growth factor, FGF. These are all paracrine factors. Many of them actually stimulate mitosis. That's the growth factor element here, is they're stimulating these cells to go through mitosis and therefore grow the embryo or grow the tissue by making more cells. And so just if you have no clue, you see an abbreviation and it happens to end in GF, you say, aha, paracrine factor. Okay, and one big family, a super family in fact, of paracrine factors are the TGF, or transforming growth factors. TGF beta is one whole superfamily. TGF alpha is a whole other set of genes, whole other set of paracrine factors, but distinct from this family. Has over 30 members, including one subset known as the bone morphogenetic proteins. And I know these names are funky, but it just was, this is a historical name based on where they originally were discovered. And now we find out these things that might have been found in a situation of studying cancer have a lot of roles to play in early development and patterning and signaling between cells. So what these guys do, they're ligands of course, because they are being secreted by the cell, and they're going to find receptors on the target cells. And in fact, this situation with the transforming growth factor family involves a double receptor of 2 different gene products that the ligand will bind to. Sometimes it's homodimers, meaning, for example, 2 of the same gene product, or heterodimers, partnering with different proteins that are encoded by genes in the same family. So I said I was going to give you a limited number of pathways. We've had the Wnt pathway. This is pathway number 2, and it's the TGF-beta signaling pathway. And so what happens here are these ligands. There's 2 different examples shown here. TGF-beta ligands binding, or BMP ligands binding. They will bind first to a receptor 1 that's a dimer, shown here in blue. And then that complex then immediately binds a second gene product, a receptor 2 in purple. And then some phosphorylation happens, some cross-phosphorylation between these proteins in the membrane. And that activates the pathway. So then the intermediate molecules are known as SMADs, and there can be slightly different ones depending on which BMP or which TGF-beta you're talking about. You don't need to know them for this class, but just know that it gets to the SMADs. And then the SMADs enter the nucleus, bind to DNA, and function as a transcription factor complex. So this brings it from extracellular through the cytoplasm into the nucleus, differential gene transcription. Same thing. Same theme. Okay, so what are the key products, key molecules within the dorsal blastophore lip? We knew about beta-catenin in the nucleus center, so what's in the dorsal blastophore lip? Because that's even more powerful and more exciting. So when this was found, when these experiments began to be done, it was like this flurry of research within the field where everyone's trying to find the factor, because you know this is going to make you famous, right? If you can find the one. So they start out with these showing experiments, try to find the genes expressed in the right place at the right time, and then take that and try to see what it can do on its own, see if it's efficient, see if it's necessary. And so here's one example, goose coin. We're looking by in situ hybridization and show you where the mRNA is located. Located right here on the dorsal marginal zone, aka the organizer. They started to do these experiments that are called heterologous injection experiments. So that would be you take this protein or mRNAs, which you could synthesize in a test tube. Using a needle, you inject cells here on what would be the ventral side, presumptive ventral side. You can see it gets expressed, and then you wait and see to what extent can you replicate that primary embryonic induction or the second body axis just by providing this single external factor. And in many cases you could get some degree of axis duplication. Here's a little blob of a thing that actually has two heads and probably some dorsal neural tube structures within that. So many of these molecules were tested. And come to find out, there is not just one. There is a lot of these factors that are expressed at this time and have some ability to replicate the body axis. So here is just a partial list of some of them. And this included some nuclear proteins that may be transcription factors. It included some secreted proteins, which might be paracrine factors. And both of these kind of make sense because you obviously need for the cells in the dorsal-blasted forelips to be able to communicate with the rest of the embryo, right? Because you have to get all these cell fates, not just in one limited group of cells, but across the whole embryo. And you're going to need some transcription factors because that information has to be received and then translated down to target genes turning on and off to establish these fates. So, many of these factors, we also realize they're actually maternally placed within the embryo. So the frog is an organism, the newt also, that relies fairly heavily on maternal components and is not going to go through the mid-blastoid transition and activate its own zygotic transcription until somewhere, around this stage. So if there are maternal products there, they would be really good candidates for setting up some of these first events that involve body patterning, cleavage, ejaculation, all this stuff. And it turns out that many of these are actually within this TGF-beta superfamily. So here is a diagram of an embryo right around the stage where it's going to begin gastrulation, and looking at some of the factors that are present in the embryo at this time that are contributing to these different signaling centers. So for one thing, here at stage 8, late gastrulate embryo, we have some factors, including a TGF-beta, which I just denoted by a star there, that are found in the vaginal part of the cell and may be involved in that mesoderm-inducing signal. So remember, vaginal has to induce mesoderm to any cells that come in contact with it. And then overlapping with that, we have beta-catenin over here in the future nucleic center cells, and we know that it's necessary and sufficient. So beta-catenin, probably in combination with some of these other maternal factors, is going to activate a gene called nodal-related. So X and R stand for Xenopus nodal-related. And it's going to be found in a very high concentration in what is the nucleic center. And then as you look across the embryo from dorsal to ventral side, this concentration of X and R sort of tapers off. So here we have a situation where you do have one single molecule that's expressed across the embryo, but not evenly. Some areas have a lot, some have medium, and some have very low. And interestingly, this having high amount versus low amount, different cell fates down the line. Organizer cell fate versus just ventral mesoderm. So this is by any means not the only factor, but it's a pretty critical one. So then down the line, one step further in time, we start to get more and more factors being activated. So I'm going to distill this now to a chance to talk about another key concept that you'll hear about throughout the whole course, so we're ripping right along here. Concepts are flying left and right. And this is the morphogenetic gradient concept. And what this involves is a single molecule found at different concentrations across the embryo. And its function is as a morphogen. And this means that this single molecule can specify different cell fates based on how much is present. So a high concentration is XNR, state number 1. Medium concentration, different information now, state number 2. And a low concentration is the third type of information, it's fate number 3. So this is nifty because you can just take one molecule and you get all this different information for the embryo from a single molecule. And the key is this difference in concentration in different spots of the embryo. So it's pretty easy for the embryo to set this up, and we can find many different examples in different species and lots of different morphogens at different times that kind of play out with this paradigm that concentration relates to cell fate. Okay. So within the new embryo at this time, we're getting actually multiple different morphogen gradients. And they actually have an antagonistic effect on each other. So over here in the cells of the dorsal marginal zone, that is the site that will become the dorsal lip of the blastopore. These cells are expressing high concentrations of a protein called noggin. And so this wedge here is meant to indicate highest concentrations of noggin on the dorsal side and tapering off as you get to ventral. On the other hand, we have BMP4, which is TGF-beta. This will be in a gradient highest on the ventral side, tapering off as you go towards the dorsal side. So we have BMPs and noggin, two opposing gradients, and as a matter of fact, noggin actually inhibits BMP4, so that even makes it a more steep gradient of, are you going to have noggin activity or are you going to have BMP activity, or some kind of intermediate stage between the two. So noggin is a parasitic factor, BMP is a parasitic factor. So all these are soluble molecules that might be, say, made in one place and could infuse gradually to some distance, but probably these cells over here in the dorsal side are making the most noggin, and then less and less is made, and less is present as time goes by, or space goes by. All right, I don't know why I had this that way. Okay, so we can take a molecule like noggin and start to manipulate it, start to look like move it or block it experiments and see what sort of properties noggin has. And based on our shown information, our in situs or immunohistochemistry that says noggin is really high on the dorsal side, it's probably associated with dorsal fate. Let's test this idea. So here, what they've done is inject these embryos with different amounts of noggin. Some got a really high concentration, some got a medium, some got a low. So if this morphogen idea is true, we should see then different cell fates develop based on the amount of noggin that got injected. And true enough, the ones that were injected with super high amounts of noggin, maybe even more than the endogenous embryo had, those became dorsalized, which means it's overrepresented for a dorsal cell fate. Okay? The ones that didn't get any noggin at all became ventralized. So, and this would, by the way, we had wiped the slate clean here by treating with UV to begin with, so we had obliterated any dorsal information, and now we're adding it back by adding in noggin, or we're attempting to. So the ones that didn't get any noggin at all is totally ventralized. And then there was this happy medium where we happened to inject just exactly the right amount of noggin to recapitulate that morphogen gradient within the embryo, and we got a totally normal patterning out of this. So you can really see concentration does have an effect here. And you can see noggin is acting as a sufficient molecule within the dorsal lip of the blastopore to set up this patterning event. Any questions? So in the sum, after a number of experiments got done, this kind of model was developed here. So on one side of the embryo, we have some dorsalizing signals. Noggin is one of them. There are some others, including cordon and follistatin. And in general, many of these are inhibitors of the TGF-beta superfamily, including the BMPs. On the other side of the embryo, on the intraventral side, we tend to have a number of the TGF-beta and BMP family members set up as morphogen gradients, and these two are kind of antagonizing each other. And so this gives then a range of finely tuned different signals across the embryo from ventral to dorsal side, where each cell can kind of just read out where it finds itself on that pathway or that gradient and take on the appropriate fate. So this is helping individual cells make a decision. Are they going to become ventral mesoderm or dorsal mesoderm? So they may have received a general mesoderm inducting signal, but then due to this patterning here within the dorsal blastopore lip and also on the ventral side, they decide ventral versus dorsal. And then another layer to go on top of this is that the cells that are fated to become ectoderm, they are also reading these signals. So there's multiple things going on here. These second group of cells are reading the signals and deciding, okay, within the ectoderm phase, we have to make some choices. We could choose to be neural, which is one ectoderm fate, or choose to be epidermis, which is a different ectoderm fate. So that gets added on too. Okay, so here's sort of a tie it together slide, a little bit redundant, but the dorsal ventral axes, when this first started to become imparted to the embryo, that was its sperm entry, right? So that was about as early as you could get, the first symmetry being broken. And then the establishment of that, the playing out of that initial sperm asymmetry initiating event continued on into the last of the stage. Some of the paradigms, the mechanisms involved would include induction and regional patterning. You now know what those are. The anterior-posterior axis, that actually didn't get started until somewhat later in time. We didn't start building the anterior-posterior axis until gastrulation initiated and we first sent some cells inside to the embryo. And then as they travel across the roof of that blastocecal cavity, they're extending the anterior-posterior axis. This is going to also involve a decision for the ectoderm to become neural versus epidermis. And interestingly, these two events, somewhat separable in time and organization for the embryo, but some of the same signaling molecules get involved. So they get reused at different times and in different places in the embryo. And that's actually another theme that we found out about as we've gotten more into the molecules that are actually being used by the embryo, is you'll see the same family members, or in some cases even the same exact gene, turn on again later in development and function as a paracrine factor to do some other patterning event like organ development long after gastrulation is done. So they seem to be just handy tools that might be able to be used in a number of different situations. Okay, so now let's leave molecules and get back to morphology for a little bit here. After gastrulation, we have an embryo that's more or less turned inside out compared to the blastula. On the outside, this embryo is now enclosed by ectoderm. It's going to start changing from being spherical in shape to being more long and rod-shaped. The anterior-posterior axis is beginning to be defined, as we said, by these cells moving in. And now we have cells in new positions, a whole layer of mesoderm touching a layer of ectoderm that would be in a good spot to have some paracrine factors to participate in induction. So this is what the embryo looks like at the end of gastrulation. And so as these signals have moved in and we're forming this new cavity called the archeneron, in fact, we do see induction occurring. So there's black arrows there that say vertical signals. These are indicating paracrine factors that are being secreted by the mesoderm, and they're going up and inducing a change in the overlying ectoderm. So this is the limited region of ectoderm, only that which is immediately above the involuting tissue. Sometimes we call this the roof of the archeneron because it's that upper portion of the archeneron in the embryo. Meanwhile, this other ectoderm down here on the vaginal side is not getting these signals. Then there's also planar signals, which means signals moving within the horizontal plane. So you may see some literally traveling around the corner of the dorsal blastic core ligament and out and upward to the ectoderm cells. It seems a more belabored way of getting a signal there, but you can find some examples of this. So, in fact, we see some of these TGF-beta and BMP inhibitors from the involuting dorsal mesoderm. These are inhibiting that roof of the archeneron, keeping an area that's going to be free from BMPs. And this actually will be information for that to take on the neural cell fate. So we said that some of the ectoderm cells, they have to decide neurome versus epidermis. And this little BMP-free region above the archeneron, that's going to become the neural portion of the embryo. Okay, elsewhere there will be BMPs, and that's going to induce the second choice, which is epidermis. Okay, so Hilda Mankel was married to another graduate student, also in Hans Schmann's lab, who also did some very famous